Hi everyone. This is the third lecture for your chapter Cell. In the last class, we discussed overview of the cell, what are its components, what makes a cell, boundaries, then division of labor, then all the different organelles, what is the difference between a prokaryote and a eukaryote. Then we also studied the two types of cells in eukaryotes, which are plant cell and animal cells. Then how there is variation and differences in sizes of the cells and the shapes of the cells. Okay. In sizes also we discussed different longest cells, largest cells, smallest cells, and in shapes we discussed a few of the shapes, commonly found shapes of cells. Fine. In this class we will be discussing specifically prokaryotes. Okay, I had already told you prokaryote meaning early or first, and karyote means nucleus. So before nucleus, okay, prokaryote cell. The prokaryotic cells include, or prokaryotes include, bacteria, blue-green algae, which is now known as cyanobacteria. Okay, blue-green algae is not an algae, it is actually a bacteria. Then mycoplasma, which is PPLO, and also archaebacteria. Fine, these are all included in prokaryotes. Prokaryotes multiply more rapidly than eukaryotes. I'll give you an example. Let us take E. coli. Have you heard of E. coli? Escherichia coli. So this is a bacteria. This bacteria doubles every 20 minutes. So if you started with one bacteria, after 20 minutes you have two. After 20 minutes you have four. After 20 more minutes you have 8 cells, and so on and so forth. So they multiply rapidly. In comparison, if we talk about eukaryotic cells, let us take a human cell. A human cell takes from 24 to 48 hours, okay, 1 or 2 days to just divide. So compared to the eukaryotic cells, the prokaryotic cells divide faster. One of the reasons for this is the simplicity. Prokaryotes are very simple cells, while eukaryotes are a little more complicated. A lot of variation is seen in shape and size of prokaryotes. Okay, a lot of variation is seen specifically in bacterial cells. Let us discuss these four basic shapes. You have the first shape, which is bacillus. Bacillus, wherever you hear the term bacillus, it means rod-like, okay, cylindrical shaped bacteria. Here you see a single bacillus, a single cylinder shaped bacteria. Okay, these are called rods. Fine, rod matlab danda, and because they look like a small rod, they are known as rods. This actually is a SEM, scanning electron microscope. Remember we studied last time. So scanning electron microscope will show you the outer surface, and these are the bacteria, bacillus bacteria, how they look from the outside, their surface. Diplobacilli means two bacilli together, and then you have streptobacilli. Okay, or streptobacillus you would have heard in different bacteria, right? So strepto means in a chain. Fine, here you find the bacillus shaped bacteria in a chain, so it is called streptobacilli. The second shape is coccus. Coccus means spherical. Now just fix this term in your head. Coccus sambdo, just think of a ball, okay, ball shaped, coccus. So coccus shaped, round shaped bacteria. Here again you have diplococci, meaning two round shaped bacteria. Then streptococci, a chain. And the new term here is staphylococci. Staphylo means bunch, okay, in a group. You must have heard of this bacteria called Staphylococcus aureus. Okay, it is a very common bacteria. It is even found in your mouth, on your hand, everywhere, on your face. So just find out if you have not heard of its name. Google Staphylococcus aureus, AUREUS. Fine. So Staphylo means a bunch. It is like a bunch of grapes. Okay, a group. So this is about the second shape, coccus or spherical shape. Then you have Vibrio. Vibrio means comma. Okay, this is a Vibrio. It is not actually like a comma, but like a printed comma, but it is more like the comma you write. Okay, a handwritten comma. If you see here, it is a little bit curved. Okay, it is sort of C-shaped. So this C-shaped bacteria are called Vibrio, comma shaped. Okay, in literature it is always referred to as comma shaped bacteria or Vibrio. And finally you have the spirillum. Spirillum are spiral bacteria. They have this small spiral like structure. Okay, so here you have spiral bacteria, which is spirillum. Fine. So these are the four different basic shapes of a prokaryotic cell. Now let us take one by one all the different parts of a prokaryotic cell. This is how it looks like. Okay, I've taken a normal bacterial cell. We will be studying the bacteria first, the normal bacteria, and then we will go into a few of the other prokaryotic cells. Fine. So here is a bacteria. The outermost covering in bacteria, which is shown here in this dark color, okay, shown here in the dark or bold line, is the cell wall. Cell wall is a tough membrane, okay, it is a tough layer. It is made up of peptidoglycan. Remember this, okay, peptidoglycan is a polysaccharide, like how cellulose is a polysaccharide. Polysaccharide means it is made up of carbohydrate or sugar. Now see the term here, peptido. Peptido means it has a few amino acids or peptide bonds present inside. Okay, so it is made up of peptidoglycan, the cell wall, and it surrounds the cell membrane. Okay, the innermost membrane, the yellow colored one shown here, is the plasma membrane or the cell membrane. Both the things mean the same. Okay, exception is mycoplasma or PPLO. Mycoplasma do not have a cell wall. The genetic material is naked circular DNA called nucleoid. Okay, actually the region is called nucleoid. Where you find this DNA, that region, that area is known as nucleoid. And this whatever the genetic material, the DNA, it is naked. Naked meaning there is no membrane surrounding it. Okay, there is no cover. It is just DNA floating in cytoplasm. It is circular. This is what is different from eukaryotes. Okay, circular DNA means it forms a complete circle like this. If this is the DNA, it will be folded and it will come back to this original place. So it is circular. When you talk about eukaryotic DNA, it is mostly linear. Okay, it is in parts. It is like this, but the ends never meet. They never meet. Fine. So this is the difference. The DNA here is circular, and the region where you find DNA is called nucleoid. This nucleoid is actually attached at some place on the cell wall. Okay, just so that it does not keep moving around, it is a little bit attached somewhere on the cell wall. Many bacteria have small circular DNA outside the genomic DNA. Okay, what is shown here in green. So this is your genomic DNA, the genetic material. Okay. Apart from this, whatever DNA you see floating around is called plasmid. One more thing, bacteria or prokaryotes do not have chromosomes like we do. Remember, chromosomes, they are X-shaped structures. Okay, they are made up of proteins called histones and DNA. So bacteria, they are haploid. First thing, if you do not know this, write it down. Prokaryotic cells are haploid. Okay, haploid meaning they have only one copy of every gene. And so the DNA that is found in prokaryotes is also referred to as chromosome. So bacteria have a single chromosome, only one chromosome. Whenever you hear bacterial chromosome, it means bacterial DNA. Clear? Right. So these extra genomic DNA, extra genomic matlab what is not included in genome, that extra, something extra. So this extra genomic DNA is called a plasmid. Plasmid is also again made up of DNA. DNA is double stranded, never forget. So the plasmid DNA, what is its use now? So it confers certain unique phenotypic characters. Confers matlab to give. Okay, it gives some unique phenotype characters, something unique to the bacterial cell. Think of it like you have made a kheer, okay? And in that kheer, in that pudding, you add a little bit of saffron, kesar. So that kesar is something extra. Eના વગર ખીર બી બને જ છે. So the kheer is normally there. But that kesar gives it something extra. It gives that color, it gives that fragrance, right? So here plasmid DNA is not really required for living, but it gives that something extra to the bacteria. What is that something extra? It could be things like resistance to antibiotics. Okay, have you taken antibiotics? Shardi thai hoy, udras, tav, infection, then doctors normally suggest you antibiotics. So when you take these antibiotics, normally what should happen is these bacteria should die. But if that bacteria has a plasmid, and on that plasmid is a gene for this antibiotic resistance, then the bacteria will survive the antibiotic and your infection will not go away. Okay, so this antibiotic resistance genes are actually present on the plasmid. Another thing is these antibiotic resistance genes you will have or must have read in biotechnology. Remember, they were the selective markers, okay, on plasmid. If you remember, very good. If you do not remember, do not worry, we will cover it. Okay, but just remember these plasmids have some genes, okay, and these genes are very helpful to the bacteria, fine, and it can fight a certain antibiotics if it has the plasmid. Prokaryotic cells do not have any organelles except ribosomes. The only organelles you find in a prokaryotic cytoplasm are ribosomes. Ribosomes are nucleoproteins. Nucleoproteins means it has RNA as well as proteins. Okay, you can see here this is the larger subunit. It is made up of two subunits. Okay, subunit is part. So it has a larger part, which is larger subunit, and a smaller subunit. The larger subunit is 50S. Here S means Swedberg's unit or sedimentation constant. Think of it like this. If you have a test tube, okay, and in that test tube, if you put this large subunit and you give it a go, okay, you swirl it around. તમે એક ટેસ્ટ ટ્યુબમાં આ લાર્જ સબયુનિટ નાખો અને એને એકદમ જોશથી ગોળ ગોળ ફેરવો so you already know that the things will settle down in the test tube, right? So it takes 50 Swedberg units to settle down. That is what is meant by 50S. And this 30S means it takes 30 Swedberg units or 30 sedimentation constant. Together, 50 plus 30, you should think, oh, it should give you 80. But no. Together, 50 plus 30 will give you 70S. Okay, so when the ribosome is complete with the large and the small subunit, it gives you a 70S ribosome. Clear? So the prokaryotes have 70S ribosomes. Ribosomes are actually factories for making protein. So you have this mRNA, messenger RNA, and the ribosome will read the message from this messenger RNA and start producing protein. Fine. You can see here, if this is the large subunit, it has two RNAs, 23S rRNA and 5S rRNA. rRNA is ribosomal RNA, and 31 different proteins. They come together to make this large subunit. 21 proteins and 16S rRNA comes to make this 30S subunit. And together, this is why it is called a nucleoprotein. It has nucleic acid, RNA, specifically rRNA, and it also has proteins. Fine. So this is all about ribosomes. They are the protein factories of the cell. Bacteria have non-membrane bound inclusion bodies. Inclusion bodies shown here are nothing but they are just granules. Okay. Try doing this sometime. Just go and take some flour, take, let us say, wheat flour or any flour, and add it to water. You will see that some flour granules, amuk lotna je kani hoy, એવું કણી કણી જેવું બની જાય in the water. Okay, it forms these small granules. Similarly, in a bacterial cell also, some of these molecules, they come together and form these granules. These granules are called inclusion bodies. Okay, they do not have a separate membrane. These inclusion bodies store phosphates, okay, polyphosphates, मतलब घना બધા phosphate जो होए, they will be stored. It can store protein, they are protein aggregates. They can also store polyhydroxybutyrate, which is a sort of fat. Okay, so they store fats. They can also store glycogen, which is a polysaccharide, right? It is a polymer of glucose. So that is what is inclusion bodies. Clear? They are nothing but aggregates or granules of different things. A specialized infolding of cell is called mesosome. Okay, now this cell membrane, the innermost brown colored thing you see is your cell membrane or your plasma membrane. Plasma membrane is not uniform. Okay, it is not what you can say, uniform. Okay, but it has some infoldings. અમુક folds છે અને અંદરની તરફ fold છે, that is why infolding. These infoldings are called mesosome. Now just imagine that scenario I gave you in the last class. If you have students from LKG, students from 12th standard, students from, let us say, 7th standard, all of these studying together in one common hall, then there will be chaos, right? Some students might be crying, some might be studying maths, some will be studying science, some will be studying social science. So it will all create a loud noise and nobody will be able to study properly, right? Same thing. Think of this cytoplasm or think of this prokaryotic cell. This cell does not have any organelles. So where will the division of labor occur? Okay, let us say on the one hand, you are breaking down glycogen to get glucose, and on the other hand, you are making glycogen from glucose. So these reactions should not take place nearby, okay, or at a time. So what happens is some reactions take place near these mesosomes. Mesosomes create that specific environment for one kind of metabolism to occur. Okay, so wherever you see mesosomes, you see a lot of increased enzyme activity. Okay, the enzymes will be found here and some or the other metabolism will be happening near this mesosome. Okay, it increases the surface area of the cell where these reactions can take place. Next is, apart from the outermost cell wall, you also have something known as glycocalyx. Wherever you hear the term glyco, without a doubt, it has to do with sugars. Okay, it has to do with carbohydrate. So this glycocalyx is present outside the cell wall. Here the cell wall is shown in this light brown color, and outside that light brown color you can see this blue color, and that blue colored thing is called glycocalyx. From the name itself, you can understand it is made up of polysaccharide, it means sugar, and glycoprotein. It means protein which has some sugars attached to it. Okay, glycoprotein and polysaccharide. Glycocalyx can be of two types. There can be two types. One is called slime layer. Slime, in our vernacular language, you call it chiknu. Okay, chiknu layer. So that is the slimy layer, the gooey layer. So glycocalyx can either be loose, as you see here, it is very loose, and it is uneven. So uneven, loose layer is called a slime layer. This is one type of glycocalyx. Another type is a capsule. Okay, capsule. Have you ever seen those medicines which look like a capsule? So here also, surrounding the bacteria, there is this outer covering of glycocalyx, which is hard, it is very tough, and it has even layer of polysaccharide. It is very even, it is very uniform. This is called capsule. Both of these, capsule and slime layer, are called glycocalyx. Okay, they are types of glycocalyx. Now, what is the role? Why is this needed? So, the role is in protection. This capsule as well as the slime layer protects the cell. Specifically, the slime layer. If it is very slimy, if it is very sticky, then what will happen? That the WBCs, which normally do phagocytosis, right, to kill bacteria, to eat bacteria, if the bacteria has this slimy outer cover, they will slip through the phagocytic cells. Okay, so your WBC will not be able to catch these bacteria because they are very slimy. Okay, they will just slip away. So that is one role. It is for protection against phagocytosis. Then second role is it protects against dehydration. It does not allow this bacteria to dry out. Okay. Then third is it helps in attaching to a surface. I will give you an example, a common day example, life example. Have you ever seen that a bucket, okay, in vernacular, dole hoy, so it becomes sticky sometimes when you touch the inner surface. After a long duration, if you have not washed the bucket properly and if it had water, the inside becomes very sticky. अंदर से एकदम चिकनी चिकनी लागे. That is because of these bacteria with slime layer. They have grown and attached to the surface of the bucket. So slime layer is also helping in attachment to the surface. And these are the different functions of glycocalyx. Okay. I hope you are learning something new. You are learning something better during these lectures. Okay, something different from what you have already learned in your common classes, right? Fine. Now, for locomotion, that is for movement, bacterial cells have flagella. Okay, they do not have cilia. They have flagella. Now, flagella is made up of a single protein fiber. We will study later on that eukaryotic flagella has many protein fibers bound together. Okay, it is a bundle in case of eukaryotes, while in prokaryote it is made up of a single protein fiber. Okay, that protein is called flagellin. Because it is making flagella, you call it flagellin. The flagella is divided into three parts. Okay, you have this basal body. This is the outer membrane, or the what you can say, this one, cell wall. Okay, the cell wall outer membrane. And so beneath the cell wall till the cell membrane, you have some structures which are called basal body or basal granule. Okay, basal meaning base. Base is where the flagella is attached. So basal body here. Then you have a hook. Hook is where the filament will be attached, where the flagella is actually attached. Okay, so this is the curved portion which is called the hook, and attached to the hook is filament. Okay, the long thing you see here, attached to the bacteria. That is actually filament you are seeing. But otherwise, there is this filament, the hook, and the basal body or basal granule, sorry. Okay, so base, hook, and filament, three portions. Apart from this, these small projections you see, okay, these small things are actually called pili or pili, depends on how you want to call it. So these pili are used for conjugation. Conjugation is like sharing DNA with your brothers and sisters. Okay, so it is horizontal gene transfer. Let us see here. So you have one cell which has a specific plasmid. Okay, this one cell has found a plasmid which had antibiotic resistance gene. What will this cell do? This cell has pili, so it will attach this pili to the next cell which does not have a plasmid. Now slowly this pili acts like a bridge. Okay, it acts like a straw, a tube, a canal, from which this plasmid DNA will be transferred to the next cell. And in the end, when the pili detaches, you now have both cells with plasmid. So this is called conjugation or horizontal gene transfer. Horizontal because you are giving the DNA to the same generation. Okay. If you give it to the next generation, it is the normal gene transfer. Okay, vertical gene transfer from one generation to next. However, if you give it to your same generation, it is called horizontal gene transfer, or in this case, conjugation. Now let us see types of bacteria. There are many ways to classify. Okay, you can classify them in multiple ways based on shape, based on size, based on if they cause any infection. But here we will study them on the basis of their structure. Okay, so based on their structure, they can be either gram positive or gram negative. I'll tell you what is the difference. Gram positive bacteria have very thick peptidoglycan layer in the cell wall. So their cell wall is very thick. Okay, so they are called gram positive. We'll see why they are called gram positive. Okay, just remember positive meaning lot of peptidoglycan and very thick cell wall. Then you have gram negative bacteria. Gram negative bacteria have very thin peptidoglycan layer or peptidoglycan cell wall. Clear? Now let us see why they are called gram positive and gram negative. There was actually a scientist called Gram, and this Gram, he discovered this staining technique. Remember this, cells cannot be seen unless and until they are stained. Okay, you have to color them to be able to see. So he used this staining technique, and it is now known as Gram staining. What is Gram staining? You take the bacteria sample, you add a dye which is called crystal violet. Okay, ask your parents in olden times when they used to have boils. Okay, જેને ગુમડા કહેવાય એવા ગુમડા થતા ને ત્યારે violet color ની purple color ની dye લગાડવાનું કહેતા. So that dye is nothing but crystal violet. Okay, ask them sometime. This crystal violet, it will enter the cell and it will make the cell violet in color. Okay, violet is just like purple. Fine. Now to fix this dye so that it does not come out. હવે આ જ ડાયને અગર તમે પાણીથી ધોવો આ સેલને તો ડાય તો ઇઝીલી નીકળી જાય. એ ઇઝીલી ના નીકળે. For that you do iodine treatment. When you do iodine treatment, this blue or purple becomes a deeper color. Okay, it gets fixed. After that you add, let us say for just now, you add different solvents. Okay, it is mostly either acetone or acetone plus alcohol. What will happen is, because the gram negative cells have very thin peptidoglycan layer, the dye will come out easily. Okay, it will be washed away very easily. While on the other hand, gram positive bacteria have a very thick cell wall, so the dye will not exit. A second dye is used which is called safranin. Safranin is a pink colored dye. Okay, So safranin, when you add, it will not dye this gram-positive bacteria because it already has this purple dye. But the gram-negative cell, which does not have any color as of now, that cell will become colored by using safranin. So you will see after doing all this process, if you see on the slide purple colored bacteria, you call them gram positive. If you see pink colored bacteria, you call them gram negative. They are pink only because their cell wall is thin. Okay. Example of gram positive bacteria are all the species of the genus Bacillus. Okay, simplest one would be lactobacillus, if you have heard, the bacteria that makes curd, right? And then you have gram-negative bacteria, and the example is E. coli or Salmonella. Now we are discussing types of bacteria. So apart from the normal bacteria, the eubacteria, you have cyanobacteria or blue-green algae. They are called blue-green algae because people used to think that they are algae, but actually they are not. They are bacteria. They have chlorophyll A. Remember, you have different types of chlorophyll, A, B, A3, a lot more. Fine. So here you have chlorophyll A and some phycobilins. Phycobilins are blue in color. Okay, they are just pigments. If you remember, well and good. If you do not, we will cover it in when we study photosynthesis. Okay. So cyanobacteria are blue-green algae and they do photosynthesis. So they are autotrophs, photoautotroph. Example is nostoc, Anabaena, Oscillatoria, Spirulina. I'm very sure you have heard of these names. Okay. So this is how a cyanobacteria looks like. It is like a normal bacteria, but apart from this DNA and ribosomes, you have some structures present here which are called thylakoids. Yes, these are same type of thylakoids you find in chloroplast. Okay, so here you see these infoldings of the membrane which are called thylakoids, and inside this thylakoid you have different molecules of chlorophyll and phycobilins which will help in photosynthesis. Clear? Now cyanobacteria can either be unicellular, so they are single in cell, or they can be colonial, okay, so a group of cells, or they can be filamentous. They form this long string-like structure, filamentous. In filamentous cyanobacteria, there are heterocysts present. These heterocysts are the cells which have given up doing photosynthesis and they do nitrogen fixing. Okay. Now, because I have mentioned nitrogen fixing, you must have heard of Anabaena, okay, and nostoc, which can actually fix the atmospheric nitrogen and make ammonia. Okay, so they are actually important in some agriculture. The last type of bacteria are archaebacteria. Okay, archaebacteria are mostly anaerobic. Anaerobic meaning they respire without use of oxygen. They live in extreme environments. Okay, so they are known as extremophiles. What are these extreme environments? Let us say a hydrothermal vent. Hydrothermal vent is garm pani ka jharna. Okay, you must have seen or heard of them somewhere. Garam pani ka kund somewhere. Okay, so basically these are the vents or places where there is hot water and steam coming out. They could be lakes, they could be ponds. So these kind of places also has bacteria, and these bacteria are actually archaebacteria. Then saline is, as you can see here, where there is a lot of salt. Okay, so much salt that normal cell cannot grow. But even so, you will find there bacteria which are again archaebacteria. You will also find archaebacteria in the Arctic. Okay, Arctic and Antarctic, you know, they are the poles. Okay, and they are very cold climate. Even in this cold climate, you will be able to see archaebacteria. So they are extremophiles. They love extreme environment. Because they are extremophiles, scientists believe they are the oldest living things on Earth. Okay, on the Earth, these were the first bacteria to have developed because early Earth was actually this kind of environment. Okay, it had a lot of hydrothermal vents. There was a lot of salinity. Fine. Their cell wall is made up of pseudomurine. If you can remember this, very good. If not, it's okay. But the cell wall is made up of pseudomurine. Example are thermophiles. Thermophiles. Thermo means temperature. Phile means to love. So they love high temperatures. So they are called thermophiles. Then methanogens. Methanogens actually produce methane. Okay. They decompose and produce methane. If you know natural gas, CNG. So where is the CNG coming from? It is coming from ground. It is coming from earth. And there are some bacteria which are found there which produce this methane. Okay. Then you have halophiles. Halophiles meaning salt loving. They love salt. Similarly, there can be acidophiles which love acidic environment and so on and so forth. Okay. So these are the extremophiles or archaebacteria. This is a diagram of very different or diverse conditions where you find these extremophiles. I will not be going through all of these because these are not important. But when I give you this presentation, you can go through this just for your knowledge and information. Okay, and you will be like curious. I can understand you are very curious to find out what all conditions can you find bacteria in. So every condition where no living organism can possibly exist, archaebacteria you will find. Some questions, quickly we will go through these. The term glycocalyx is used for a layer between present between cell wall and membrane. No. Cell wall of bacteria. No. Bacterial cell glyco engineered to possess N-glycosylated proteins. No. A layer surrounding the cell wall of bacteria. Yes. Glycocalyx is present outside the cell wall. So the answer is D. Which of the following is not found in prokaryotic cell? Nuclear envelope? Yes, A is the answer. Okay, ribosome is found, then mesosome is also found, and plasma membrane is also found. What is not found is nuclear envelope. The DNA is naked. Pigment containing extension in some cyanobacteria are. So in some cyanobacteria, there are pigment containing structures. These structures are heterocyst, basal bodies, pneumatophores or chromatophores. The answer is chromatophores. Chroma means color or pigment. Okay, so from the name itself, you should be able to tell. Heterocysts are the cells which do nitrogen metabolism. Basal bodies are found at the base of flagella. Pneumatophores are nothing but they are roots of some plants which come out of the soil. Okay, and they grow negative geotropism. They grow upwards. So these are called pneumatophores. Right, so the answer is chromatophores. Next question. Which among the following are smallest living cells known without a definite cell wall? So these are the smallest cells and they do not have a cell wall. They are pathogenic to plants as well as animals and can survive without oxygen. As soon as you hear this, smallest living cells and without a cell wall, the answer is always mycoplasma. But it is my job to tell you that yes, mycoplasma are actually bacteria which cause infection and disease in plants as well as in animals. Okay, and because they are such parasitic, what you can say, parasitic or infectious bacteria, they do not really require oxygen to survive. The answer is mycoplasma or PPLO. Next question. Which of the following component provides a sticky character to the bacterial cell? What provides this stickiness to the bacterial cell? Cell wall, nuclear membrane, plasma membrane, or glycocalyx? The answer is glycocalyx, specifically the slime layer. Okay, the slime layer type of glycocalyx. Select the wrong statement. Bacterial cell wall is made up of peptidoglycan, that is true. Pili and fimbriae are involved in motility of bacteria. No, pili is actually used for conjugation or attachment, okay, attachment to different surfaces, but it is not used for motion, for locomotion. So the answer should be B. Cyanobacteria lack flagellated cells. True, cyanobacteria do not have flagella. And mycoplasma is a cell wall-less microorganism. That is also true. You had to pick out the wrong statement. Next question. Which of the following is not a feature of plasmid? This question is relatable in this chapter, cell, as well as in biotechnology. Okay, so it is a common or it is a connecting type of question. Plasmids are circular, yes. They are transferable, yes. We saw conjugation. They're single-stranded, no. They are DNA, and DNA is always double-stranded. Do they replicate independently? Yes. This we will study when we study biotechnology, or you might have already studied and you remember. Right? So the answer here is single-stranded, which is wrong. Which of the following are not membrane-bound? Vacuoles, ribosomes, lysosomes, mesosomes. The answer is ribosomes. Okay? Ribosomes and centrioles are the organelles which do not have a membrane. Which of the following is not an inclusion body found in prokaryote? Phosphate granule, it is an inclusion body. Cyanophysae granule, it is also an inclusion body. It is basically a dye, okay? Cyanophysae. Cyanophysae. Then glycogen granule, also found. Polysome, no. Polysome is actually multiple ribosomes attached to one mRNA, but polysome is not an inclusion body, so the answer is D. Motile bacteria are able to move by Fimbriae, flagella, cilia, pili. We again come across the same type of question, okay? The answer here is flagella. Motile means the bacteria which can move around, okay? Motion, like mobile. Same thing, motile. So motile bacteria are moving around because of flagella. What is the name of region where double-stranded single circular DNA is found in prokaryotic cell? The answer is nucleoid. Nucleoid is the region where you find bacterial or prokaryotic DNA in the cell. Fine. The two domains to which prokaryotes are classified into are, in the 5-kingdom or 3-domain classification, you have prokaryotes which can be classified in 2 types, okay? 2 domains. So which are these? The answer is bacteria and archaea. We saw both of these are prokaryotic cells, okay? Bacteria, yes, but protista, no. Protista are eukaryotic unicellular cells. Archaea, again, yes, but eukarya, no. Eukarya, no, but Monera, yes. Monera is just another term for bacteria. Clear? When a water sample from a hot thermal vent was isolated, it was found to contain a single-celled organism having a cell wall lacking a nucleus. What is its classification most likely? From the first line itself, you know it is found near hot thermal vent, so it is an extremophile. An example of extremophile is archaea, right? It is single-celled, again a prokaryote, has a cell wall but does not have a nucleus, so it is definitely archaea. Okay. In prokaryotes, the hair-like outgrowth which attach to the surface of other bacterial cells are pili. Okay. To attach to another bacteria, you require this conjugation pili or sex pili. Okay, and this is the answer. So there we end today's class. I hope you had fun. We completed almost everything that you should know about a prokaryotic cell. Fine. See you next time.